So what I'm going to talk to you today is some of the stories we've learned from laboratory and discovery research, and also partly preclinical research on the scope of the cancer immunotherapy, combination therapy. So it's too loud. Okay, great. So let's say in the last couple of years, and we've witnessed many excitement in the field of cancer immunotherapy, particularly those approval of some of the antibodies against the immune checkpoints, such as CTLA-4, such as the PD-1. But yet, we are not there yet, because as we learned, it's only a subset of the patient would respond, would benefit from those checkpoint blockades. What we also learned in the clinic is apparently there's two finally defined tumors can be diagnosed. So on the left side, you will see most of the small subsets of patients actually would be seen as a lot of persisting T cell infiltration already in the tumor. At the same time, you would also see there's many chemokines already being abundantly expressed in this tumor, which allow them to attract the leukocytes. And then at the same time, we also see many immune regulatory factors, for example, PD-L1, IDO, and FOXP3 also involved, which provide a kind of suppressive signals to the tumor to block the effect function of the T cells. So many of the clinical data has suggested that this kind of patient would mostly respond to a single agent immunotherapy. However, the majority of the patient, as you see, will be on the right hand, they don't have any T cell infiltration. And this patient, usually they do not respond to the immunotherapy. So a huge task ahead of us is how to convert this non-inflamed tumors into inflamed tumors, and to convert these non-responders to the responders. So our colleagues at Genentech, and Dan Chen and Melman, a few years ago, has drawn a very nice cartoon illustrating the many steps in cancer immunity cycles. So basically, what you need to achieve in an antigen-specific T cell response is you need to have this professional antigen-presenting cells, including dendritic cells, to be able to take up the antigen which are released from cancer cells. Then once they activated, they are able to migrate to singular lymphoid organs where they prime the T cells, and they educate the T cells to become the effect function T cells, and then to allow these T cells to migrate back to the tumor to execute their killing activities. So this provides us a lot of opportunities to really harness the power of the immune system to achieve a better attack or better anti-tumor immunity. So as illustrated here, you can even think of using the conventional therapy, the chemotherapy or radiotherapy, to enhance the release of the cancer antigens. And then there's multiple pathways you can provide additional signals to activate dendritic cells. And then additionally, most important is to really provide many co-stimulatory signals to help the T cells to become more effective function T cells. So among these, I'm going to talk one samples which have to deal with the activation of the dendritic cells. It is a very old molecule. It's called a CD40. It's been identified, I guess, more than 20 years ago, initially identified as a receptor on the B cells, then lately was found to be expressed by many of the myeloid cells, including dendritic cells. So what's important is that when dendritic cells get activated, they upregulate CD40, which would activate the antigen-specific T cells. Then the T cells, in turn, would provide many of the so-called licensing factor. One of them is CD40 ligand, and another one is the lymphotoxin. So these two licensing factor would then feed back to the DCs and to help them firstly upregulate many additional co-stimulating molecules, for example, CD70 and the 4-1BB ligand, OX40, which provide a ligation to the receptor on the CD8 cells. At the same time, also there's many effector molecules being released because of the ligation of CD40 ligand, for example, interleukin-12 and the type 1 interferon, which induced by the lymphotoxin. So we've had anti-CD40 antibodies, and just here's an example to show you in vitro, and this antibody does activate the disease. For example, as shown here, the upregulation of CD83 and the CD70 on the monocytes-derived disease. So this molecule has been in the clinic a few years ago, and this is the phase 1 study. We had a combination of CD40 and gemcitabine. And so the left panel indicates that actually among all the patients tested, and almost half of them responded quite nicely. And on the right side, and there's a representative of one patient, you really see a shrinkage of the tumor, both the primary tumor and also the liver metastasis has been really shrinked to a smaller size after 3 cycles of the treatment. So preclinically, we developed immunized anti-CD40 antibodies, and this antibody is also very active in the mouse, which is able to activate dangerous cells. For example, just looking at the upregulation of CD40 and CD86. So this is by the IV injection. It's also working on subcutaneous injection of the anti-CD40 antibody. Basically, you see a similar thing. Then if you track in a draining lymph node, you really see the activation of dangerous cells has been popped up in these draining lymph nodes, as indicated by the MHC high and C11C high populations. So, but then what we also realize, if you look carefully, for example, here in the spring, after treatment with anti-CD40, we also see an increase of the T regulatory cells in the spring, which is not something you like to have, because this might provide a hampering of the immunity. So then we did a careful study in many of the tumor models. What we do realize, so this is in the orthotopic models where the pancreatic tumor cell lines were injected in the pancreas. So we start treatment after 7 days after tumor being implanted. Then you really see with CD40 antibody alone, you see initial drop or prevention of the tumor volume to be really, you see a nice inhibition by CD40. But then by time, the tumor start to grow out. So then we took a look at many time points of the tumor. What we realized is this growing out tumors, actually they are filled up with a lot of macrophages. So we call them tumor-associated macrophages. Or there's also some of them we clearly see the infiltration of regulatory T cells, which help the growth of the tumor. So then we thought, okay, we need to come up with a strategy to help these tumors, hopefully to achieve a synergistic tumor prevention. So for example, if we can remove the tumor-associated macrophage here. So we developed a very powerful antibody called a CSF1R receptor antibody. It's a dimerization receptor inhibitor. So against the CSF1R is expressed by many of the myeloid cells, including macrophage, but also including the dangerous cells, and a subset of dangerous cells, and also many of the myeloid-derived suppressive cells. So, and this antibody, again, has been in the clinic. So, and this is data we published last year, and which show you in one of the ovarian cancer patient, after treatment with 2 infusion of these antibodies, and you really see the majority of the macrophage disappeared. So here, looking at the staining of CD80, CD163, and CD68 double positive cells, and then here looking at the CSF1 receptor expression. So we also look into the many different cancer patients. So in many of them, we really see a clear the decrease or disappearing of the macrophage, indicating that our antibody is kind of specific to deplete many of the myeloid cells, particularly the macrophages. So then we move back to our preclinical models. Here I'm showing an example of the MC38 colorectal cancer tumor models where the tumor is injected subcutaneously. So after treatment, basically here looking at here, the 2G2 is our mouse surrogate of the anti-CSF1R receptor antibodies. So simply after 2 times of treatment, we really see the majority of the F480 positive macrophage are disappeared. Of course, you do still see some of the MDSCs are still there, but looking at the total TAMs, they're basically all gone. And then we also looked into orthotopic models with pancreatic tumors. And then again here, it nicely shows that our antibody is able to effectively deplete many of the macrophages. So what will happen if we combine this CSF1R antibody and CD40? So we did 2 experiments, and just to let you know, don't be confused with the color because we have different color coding for 2 experiments. So these 2 experiments are done in 2 labs by 2 different groups and in 2 different countries and in 2 complete different models. So on the left hand, you will see orthotopic models, breast cancer tumor models. So the tumor cells were injected into mammary and the fat pads. And here, looking at the percentage of mice, they are being killed when the tumor reached to above 700 cubic millimeter. So you really see, basically here, either CSF1 antibody or CD40 antibody alone had no effect as compared to the vehicle group, but the combination really significantly the mice to develop a big tumor. And on the right hand, we see also looking at the percentage of survival. So here, CD40 alone does have some effect, but the combination does much better job. So what happens to this tumor is we looked carefully into the tumor infiltrating T cells. For example, here we're looking at also here the data on the spleen. We also had the data on the tumor. So here, just measuring the effect function molecules on the T cells, looking at the CD4. Basically, what you see here is CSF1R antibody alone had no impact on the T cell function, but the CD40 certainly increased the gamma secretion and also a little bit granzyme B on the CD8. But the combination, you really see a synergized activation of these effector molecules, both for the interferon gamma and the granzyme B. So this is summarized data. You really see the combination would significantly enhance the interferon gamma producing and the granzyme B producing both CD4s and the CD8. So if you look further, and we also see actually combination increase even more of the effector memory CD8 cells as compared to the CD40 alone here. So looking at the right population here. And then we even looked into the ratio of CD8 effector memory versus Treg, surprising to us. So we really see the combination almost doubled amount of the ratio of CD8 versus CD8 effector memory versus Treg as compared to the CD40 alone. And we also confirmed that and in the combo group, the majority of the proliferating CD8 cells are CD8 effector memory cells, which are all KL67 positive. So currently, we are expecting to have the phase 1 trial for this CD40 and the CD1 combination starts this year. And currently, there's also other studies which in the combination with CSF1R is ongoing. So for example, we have a study combined with anti-PD-L1 antibody. It's called antizolizumab. So this antibody basically going to be approved soon, probably in the summertime. And another study is deal with the combination of CSF1R antibody and the chemotherapy. So stay tuned. We're going to have more data to be announced later in the coming few years. And then now I'm going to switch gear and talking about something else and more looking into the vaccine side with different adjuvants. The adjuvants I'm going to talk about is the common gamma chain cytokines, interleukin 12 and interleukin 15. Interleukin 21 and interleukin 15. So what are the goals for therapeutic cancer vaccine? I think what we like to achieve is really to increase the frequency of the effector cells through either the priming or through the rescue or amplification of the pre-existing CD8 T cells. And then we like to really elicit that the CD8 cells are able to traffic into the tumor and persist in the tumor, which potentially can provide a prolonged anti-tumor response. Then eventually also what we hope is this CD8 cells which are elicited are able to live longer in the tumor. So I mentioned, okay, I'm going to talk about two cytokines which belong to the family of common gamma chain cytokines, interleukin 15 and interleukin 21. So in the past, both cytokines has been used in clinical trials as single cytokine therapy, but because of the, I guess for the interleukin 15, because of toxicity issues, so it was had some difficulties in developing these cytokines as a drug. So we had a different idea just by accident. What we found is interleukin 15 and 21, if you put these two cytokines together, we really see a dramatic effect. Basically, the combination programs all the lymphocytic cell lineage to proliferate and also to turn on the granzyme B. So as depicted here, you really see the CD8 cells. So here looking at the CD4 proliferating cells and granzyme B positive cells. And then even for B cells, you really see all the B cells start to turn on granzyme B. Of course, we did a nice titration, and we also look at the single cytokines. We really see the combination and give a synergy in promoting the granzyme B producing proliferating lymphocytes subsets. So then we thought, okay, well, what if we combine this and to be used them as adjuvants for vaccine? So we started a very simple experiment, use very old peptide, it's called TRP2, which is a melanoma specific peptide, and we combined the poly-IC as initial adjuvant. So then we given our cytokines as adjuvants 3 times, start at day 0, and then day 2 and day 3. And then we looked at day 7 to see what's happened to the T cells. So here we use the recombinant proteins. So what it did show is really see an increase of the B cell lymphocytic lineage in the spring, so including the B cells, including the NK cells and CD4, CD8. So when you compare the vaccine alone versus the vaccine plus the interleukin 15 and 21. And then if you look carefully, what we see is the vaccine really enhanced the composition of the T central memory cells, which is CD44 positive and CD62L double positive population. So compare here, you get 10% by vaccine alone, but then you will see a doubled amount of the central memory cells has been induced, but with additional interleukin 15 and 21. And this is a summary of the data. And we really see the combination of the two cytokines driving the expansion of central memory T cells. And then we also discovered that actually the combination of the two cytokines really driving a specific population which express uniformly the EOMES and the KLRG. Also, of course, you also see the KLRG single positive cells, but the double positive population is really dramatically induced both for the CD4 and the CD8 with the additional interleukin 15 and 21. Then we thought, okay, let's move it into more applicable approach, because what we use here is the recombinant cytokines. The half-life is very, very short, so we had an idea, say, okay, let's put them into a DNA plasmid and to enhance the half-life and give them really a systemic exposure of the two cytokines. And then at the same time, we made a plasmid against one of the prostate antigens called SPAS1. So we did a similar approach, and here, but we given basically a boost of the vaccine and with additional cytokines injected by the electroporation. So this data basically confirmed what we see in the TRP2 peptide vaccine. With additional interleukin 15 and 21, and it drives the expansion of the EOMES and KLGR double positive population. And also importantly, and also induce the release SPAS1 specific pentamer positive CD8 T cells. Thanks for ProImmune provide a nice pentamer for these molecules. So I guess to conclude, and what I've showed you is really there's a lot of opportunities in the field in the cancer immunotherapy, and we should feel very fortunate to have a really wealth of opportunities. I guess really what we're hoping in the next couple of years, and we might be able to see a really revolutionary shifting in the field of cancer, in the way of cancer being treated. So let's keep an eye open, and let's hope for the best for the patient who are really suffering in this field. So to conclude, I'd like to really thank the people who did the work. So John, Max, and Caroline, they initially involved in the combination study with CD40 and CIS1R, and all the cytokine studies were done by one of the postdoc Ines in my lab. So hopefully we can make a nice publication soon this year. And there's also people from different development team were involved in this kind of study. So thank you very much.